क्या पागल। हाँ बैटरी हो गया, इसे छोड़ दो।
Hello. Hello, Yadu. Hello, Dr. Yadu. Are you there?
Yes, sir.
Yeah.
Yes, sir.
I think Dr. Islam has already joined, so you can just make him a presenter also.
I made
Okay.
presenter, sir.
Okay.
Yes,
Okay. So we'll wait for another 5 minutes, then we will start the program. Okay.
Okay.
Okay, that's fine.
Okay.
Why am I doing? আমাকে কিন্তু মাঝখানে একবার চা খেতে এমন না করো না কিন্তু।
Hello, Dr. Yadu. Hello.
Yes,
Hello.
sir.
Yeah,
Yes, sir.
now I think it's already 3:30 is crossed, so you can just initiate the proceedings for the webinar.
Okay, sir. Okay, sir. Okay, hello everyone, and welcome to today's webinar organized by School of Pharmaceutical Technology, Advanced University, Kolkata. The title of based screening of database for lead-like molecules application in SERS-CoV-2. Today's speaker is Dr. Muhammad Ataul Islam. He is a Newton International Fellow, University of Manchester, UK. I would like to invite Dr. Partho Roy, Associate Professor of SOPT, to give a brief introduction about the speaker. Over to you, sir.
Thank you, Dr. Yaduranjan, for inviting me. I hope this would be a very entertaining for the students. Especially, there are many students who are opted for CAD as an elective subject, and as a whole for the whole university, I think, as well as the other participants, it will be a very entertaining and very knowledge enriching program. So, as apart from Dr. Ataul Islam's academic achievement, he's very much known to me. We used to be very good friends during our PhD days. And informally, before introducing him, what I can say to the, especially to the students, is that he was our one-stop solutions, especially when we had any problems with our manuscripts, figures, papers, or anything. So he was the one-stop solution. We used to go to his lab to solve our queries. So apart from that, he is now the Newton International Fellow in University of Manchester, and before that he was in University of Pretoria as a postdoctoral researcher. Before that, he was been engaged in academics in, I think, Bharatiya Vidyapeeth University, Pune. So he was assistant professor over there. And before joining Bharatiya Vidyapeeth, he was honorary research fellow in University of KwaZulu-Natal in South Africa. And as I know, Dr. Islam did my PhD from Department of Chemical Technology in University of Calcutta. So this is a brief introduction of him. I hope the students, as well all of the participants from SOPT as well as from other institutions who have opted to register in this program will have a good enriched session of computer-aided drug design, and especially its application in COVID-19. So thereby I invite Dr. Islam to share his presentations and start the webinar. Thank you, Dr. Islam. Thank you for taking our invitation to give your enriching thoughts to the audience of SOPT.
Dr. Maiti, Dr. Yadu for their invitation and opportunity to present my work in this platform. So, so it is shared. Is it visible? Hello, am I audible?
No, sir. No, sir, it's not visible.
I think if you, yeah, the screen is coming. Yeah.
Yes,
Right.
yes, now it's
Okay, I just okay. Okay, so it is visible right now, the presentation?
yes, sir. Yes, sir, visible.
Okay,
Yes,
thank you. So basically today I would like to introduce about the computer-aided drug designing, and nowadays it is well known as the Pharmaco-Informatics. So how the Pharmaco-Informatics and how the pharmacy people can engage to explore the lead-like molecules or promising molecules from the database, and further that can be optimized using the experimental validation or experimental approaches. So that I'll try to explore a little bit. Then I'll show you some applications in the recent COVID-19 work. So this is a very brief overview of my today's presentation, I will discuss about the drug and drug discovery and what is the pharmaco-informatics. Definitely, I will explain. And then, although this is the pharmacophore and de novo design, these are not the scope of today's talk. But that is this is relevant because this is these two are very important part of computer drug designing. So basically, today's lecture will be based on molecular docking and how the molecular docking can be implemented to screen the large chemical databases. And finally, I'll show you 3 applications on NSP 15 inhibitors, another one the TMP RSS2 inhibitors, and finally the spike and NSP 15 inhibitors. So let's start. So this is a very well known what is the drug. I think you people know better than me. There is any chemical that influence biological process when introduced in the body, and drugs do not introduce novel physiological responses in the body of the brain. Whatever, this is the small molecules or change the chemical pathway or biological pathway. So this is the related, I thought this is known as the doctrine of signature. So that is said that God has created all diseases, definitely. And he also created an agent or a drug for every disease. They can be found everywhere in nature because nature is the universal pharmacy. And God is the highest ranking pharmacist. So this is absolutely true. So now I'm going to explain the complexity or the problem in the drug discovery and development process and how this the computational approach or the informatics help to accelerate, help to enhance the drug discovery pipeline. So if you see this is a very normal traditional drug discovery process. So first of all, you have to identify the disease and then the target identification or the target selection. So here also the bioinformatics or different experimental approaches are helpful to identify. Once you have the target, then you can go for the small molecule identification, like lead. This is called a lead discovery or the promising molecule discovery. So here we can use the high throughput screening, different types of assays, etc. And then we need the medicinal chemist expertise to optimize the molecule, synthesize, then structure activity relationship, then further modification, and so on. Once we have the molecule in our hand, then we can go for the experimental studies like in vitro or in vivo studies. And finally, we can go for the preclinical trials or like clinical trials. And then there are a lot of approvals. So this is not a simple process. This is a very much trial and error basis method, and in any stage you may fail. So till now the success rate of the drug discovery is absolutely low, that is less than 1%. So if you start from 10,000 or more than that molecules, so may have finally you get one molecule or may not be. So what is the actually, suppose here I'd like to explain how that is if you start from 5,000 to 10,000, maybe you consider more. So you can apply the different target identification or assay development or lead generation. So that may take about on an average 3 to 5 years, depending upon your expertise, depending upon your infrastructure. Once you have some few molecules, you can go about, suppose for example 250 or 500 or 1000 molecules, then you can go for the experimental validation. So it may take 1 to 2 years, and then there is phase 1, phase 2, phase 3, so on, and then finally you may have, if you are lucky, then you may have one molecule in your hand, and then go for the post market surveillance and then the market. So this is on an average it will take about 15 years of time, if you roughly you estimate 10 to 15 years, maybe more. So this is very time consuming, and also you need the huge resource, and you also need what I can say money. So in this situation, we need some smart techniques which can help the drug discovery process and reduce the time taken, reduce the cost. So in this situation, the pharmacoinformatics or cheminformatics or bioinformatics or computer chemistry. So these are actually related field. So bioinformatics basically the biological informatics, cheminformatics is basically the more oriented to the chemistry. So I would prefer the term pharmacoinformatics is very much helpful in this situation. So what is pharmacoinformatics? Very briefly I will explain here and then I will go. So the pharmacoinformatics basically two terms, the pharma plus informatics. When the informatics or the computational approaches involved in the pharmaceutical sciences or pharmacy to identify the drug molecule, better I can say the lead molecule, to data acquisition, collection of the data, storage of the data, analyze the data, and finally dissemination data. So these are the very preliminary concept of the pharmacoinformatics, that is what we can do using the pharmacoinformatics. So next slide I will explain a little bit more about it. So how the relevance of the pharmacoinformatics to the pharmacy or the pharmacy people, the students or the researcher. So this is one of the paper it published 2017. How this pharmacoinformatics is important for the bachelor's and master's of pharmacy. So they explained, I read out it actually line by line. In the early 1970s, pharmacists were not involved in direct patient care. However, soon after the introduction of clinical pharmacy, pharmacists played an important role in providing drug therapy and the patient care. Similarly, the pharmacy informatics or the pharmacoinformatics was introduced to ensure pharmacists, very important here to note down, the pharmacists are well trained not only to deliver quality care to patients, but also to aid in achieving optimal patients' health outcomes. So how can I define it? So it can be defined as the scientific field that utilize a systematic approach to process and utilize medication related data and information, including its acquisition, storage, analysis, and dissemination in the delivery of optimal medication related patients' care and health outcomes. So these are actually very, very important in case of the how this pharmacoinformatics fields is related to the pharmacy. So I would like to point out that the pharmacoinformatics is a very huge subject. So you can apply in a different way. You can apply it to data collection, data analysis, and reuse the data, or you can use that is the drug discovery, that is called the computer-aided drug design. So I would like to explain today how this pharmacoinformatics approaches applied in computer-aided drug designing or drug discovery. So these are the well-known informatics field like neuroinformatics, immunoinformatics, genome informatics, then ADME informatics, toxicoinformatics, cheminformatics, bioinformatics, and pharmacoinformatics. So all these informatics are helping to develop or to identify the drug molecule. And then you can also have drug, then we can use a different another type of several types of informatics like pharmacy informatics, drug informatics, nursing informatics, biomedical informatics, and healthcare informatics to proper utilization of this drug. So the informatics is before the drug, after the drug. If your drug is in the market, how this drug actually affecting in the trials or in the patients, that can also be assessed using the informatics. So here the pharmacoinformatics actually helped by other subject like cheminformatics, computational chemistry, computational biology, bioinformatics, and etc., and the medicinal chemistry obviously. And then these subjects when come close to each other in the pharmacy are involved in the informatics, and that actually deliver the drug in the very first way with the less animal sacrifice, with the less investment. So this is actually the very brief about the pharmacoinformatics. Now how the pharmacoinformatics or computational approach are in the drug discovery. What are the fields? So you can use in the machine learning approach. So you can go for the statistical pattern recognition. You can go to model development and the biological system analysis. You can go for the data retrieval, that is the data mining. So you can use data extrapolation, data analysis. You can go for network analysis, go for transcriptional, protein comparison, the target similarity assessment, prediction of the ligand cross-reactivity, and very importantly, binding site detection, identification of the active site where my ligand will go, where my drug will go and bind. So that is the detection of the putative binding pockets, ligandability, how my ligand is potential enough to a particular target, and obviously the docking, the protein docking, protein ligand docking. That is the two different molecules when combined to each other. So that will I'll discuss detail about docking today, later. So now I explained the importance of the pharmacoinformatics, where we can use the pharmacoinformatics. Now I'm going to explain, that is, what are the types and where we can use, how can we use, and which situation we can use the different approaches of the drug designing or the pharmacoinformatics in drug discovery. So there is four scenario here. So definitely I'll when I talk about the ligand, so that is definitely the small molecule, and we're actually talking about the receptor, that will be the macromolecule. So here, if your protein structure known, that is target known, and also the ligands are known, so structure-based drug designing, like protein ligand docking. If your ligands are available, that is ligand crystallized, biologically evaluated, but there is no three-dimensional crystal structure of your protein molecule available yet, so you can go for ligand-based drug designing, like similarity searching, Pharmaco-for model, QSHR, etc. Suppose you don't have, you have the protein structure known, that is crystallized, but you don't have any small molecule or the ligand. So in that situation, there is an option to de novo design based on the protein structure. You can design your molecule. That is the de novo design. And in such a situation, if you don't have any ligand known or the protein known, then there is no option for the computer drug designing. You have to go for the experimental approaches. So now I'll explain about the ligand-based drug designing approach. There is a pharmaco. There is a lot of approaches are there. Among them, I explore one, the pharmaco modeling. Though this is actually not relevant today's talk, but I'll explain a little bit because very essential how this ligand-based approach important for the pharmacoinformatics. So Pharmaco-for model, Pharmaco-for can be defined as the important groups or the functional groups, mainly if I say, that involved in the binding at the active site. So their relative positions, their orientation, structure orientation, how they arrange in the three-dimensional space, that is actually very much important to find out for the binding analysis. And for this, we need to know the active conformations. Also important, it is for drug designing and the drug discovery. So here you can see, if the green portion, this is the active site. So active site amino acid actually present the number of functional group. So we have to design such a molecule, or you have to find out such a molecule, so that molecule can form the potential interaction between the ligand and the protein. So suppose I'm going to explain one example very briefly, that morphine. So this morphine is important for the analgesic activity. So in morphine, this is the red portion, the hydroxyl group, methyl group, and here the nitrogen. These three groups important for this active analgesic activity. The remaining portion of this molecule are important to hold these three groups in such a orientation so that it can exert or exhibited the analgesic activity. So so if I say these are the three another two molecules, metagocine and the levorphanol. So these also contains the same set of groups and important for analgesic activity. So if I say here, so these are actually here. So now I'm going to these all the molecules. You can see this these three groups present in all the molecule in a similar way. So here I will explain about the 3D pharmacophore, how the pharmacophore model can be developed. So if I see the same example, so suppose this is the hydroxyl group, this is the methyl group, and here green is the nitrogen. So the aromatic ring, hydroxyl group and nitrogen. So we connect these things to identify or to extract their relative position or the orientation at the three-dimensional space. So like this, and then we calculate the distance, and then we calculate the angle. So this is the actually you can say this is the pharmacophore model for these three compounds. So from these three compounds, all our functional groups are not important for the analgesic activity, but these three functional groups are very crucial for analgesic activity. So we have to find out how they arrange in the three-dimensional space, and that is the purpose of pharmacophore modeling. So also we can identify the pharmacophore model from the binding site. Suppose this is the binding site, and binding site the different functional groups. So we have to design a pharmacophore contrast to these available functional group of the active site amino acids. So these we can see the basic positive or basic or positive center here, hydrogen bond acceptor or maybe donor, and this is the aromatic ring. So if we come here, just take out this information and we connect it, calculate the distance, calculate the angle. So that actually gives you the pharmacophore model. So this, once you have the pharmacophore model, so you can ask the chemist to synthesize such a molecule which contains these type of pharmacophoric features or functional groups in that molecule. So if we generalize this, the hydrogen bond acceptor or hydrogen bond donor, van der Waals interaction, ionic interaction, these are the common pharmacophoric features important for the pharmacophore analysis. Okay. So now, how we actually develop the pharmacophore model. So we consider the set of small molecules. We divide the training set and test set. Training set used for the generation of the pharmacophore model. Test set used for the validation of the model. So these are the training set, test set division, and once we have the model from the training set, we can validate this through the test set and other parameters like feature randomization test, decoy set, maybe more. And then we approve this pharmacophore model, and we can use this pharmacophore model to design the new molecule, or we can screen the database. So in short, this is the ligand-based approach. We need the known small molecules, and once we have the model, we can use the design new improved molecule, predict the biological activity, etc. So these are the different tools we can use for pharmacophore model. Now I'm going to the molecular docking. So this is basically the structure-based drug designing. So we need the structure, three-dimensional coordinate of the target, that is the receptor, and we need the structure of the small molecule. So here, if you have a set of ligands, the multiple ligands, 1,000 ligands or millions of ligands, and you have the receptor. So based on the active site, you can sort out what are the molecules in your data set are favorable for binding, are important for binding this receptor, or what are not. So that we can actually differentiate. We can separate out between the more affinity binding molecules to the less affinity binding molecules, or more active molecules to the less active molecules. So this is actually basic molecular docking. We have the ligands, you have the receptor, you use the docking engine, and you can feed the two molecules. So here the pose, I'll explain in the next slide. The pose, that means once a ligand, suppose a single ligand bind at the active site. So during the docking, it is actually conformation analysis are going on. So for a single molecule, maybe multiple of conformation generated, and it is try to find out among these conformation which one is the best orientation, or which position of this ligand is more prone to this target. So these multiple conformation at the same active site, these are known as the poses. So molecular docking or molecular docking engines identify the best pose or the best orientation or conformation of each molecule. So it predicts the pose. And also it actually gives the score. So I'll explain how we score and what are the scoring functions in later slides. So binding site, you obviously know where the ligand binds in the active site, in the receptor molecule. And it is basically generally the cavity, or that is also I explain what is a pose. That is, once we have the docking, the single molecule may have the multiple conformation. Each conformation is known as the pose, or binding mode actually. So now I am explaining the focus docking and the blind docking. Have to identify, to know, that is my and where to bind and where not to bind. So there are two situations there here. So one, if you know your active site, so that is very good. So you identify and you tell your ligand, your small molecule, that is you have to go and bind there only. You no need to search anywhere, anything else. So that is called the focused docking, or it is also known as the targeted docking. In another situation, if you don't know the exact binding site where your ligand will go and bind, so in that situation, a pocket may have the multiple active sites, and there's a possibility your ligand go and bind in any pocket. So you leave your ligand, go and find a suitable position or suitable binding site according to the ligand and bind it. So that is called the blind docking. That is, we don't have pre-information of the active site or the binding site where my ligand should bind. So this is called the blind docking. So these are the different types of docking. Now I am going to explain another types of docking, like rigid docking, semi-rigid docking, and flexible docking. So rigid docking, when your ligand fixed, receptor fixed, you are not changing the conformation of your ligand as well as your receptor. So this is similar to the lock and key. As it is go and bind. There is no conformation analysis in either in protein or in ligand. So this is called the rigid docking. So especially the protein-protein docking. This is a rigid docking, absolute rigid docking. Because it is very difficult to do the conformation analysis of the protein or macromolecules. The semi-rigid docking, here the ligands are flexible, receptor are rigid. So your ligand will go at the active site. It will there will be no conformation analysis of your receptor, but the ligand will go the conformation analysis of the sampling, and it will generate huge number of conformation. Among this conformation, it will find which one is the best one, which pose is the best one. So how to select the pose, that I'll explain in the next slide. So this is called a semi-rigid docking. The ligand is flexible, receptor is rigid. And it is the final of the flexible docking in case of the both receptor and the ligand are flexible. But here, just a minute. So in this situation, because in a real system, both the ligand or the receptors are in flexible. They are changing the conformation. So it is our drawback, that is till now there is no software package or any tools which can generate the conformation analysis of the receptor of the whole protein. So in that situation, what we do, we can give some flexibility around the receptor site. So this is, and then we go for the conformation analysis of ligand, and then we generate the docking. This is called the flexible docking. So generally, if I say most of the cases, maximum people do the semi-rigid docking. Now that I told you, that is how can we assess your molecular docking results after docking? How can you assess which pose is better, which orientation is better? So there is a two parameters. One is the binding interactions and the scoring function. So binding interaction is very important. How your ligand bind the active site, because depending upon the binding interactions, your protein ligand complex stability will be achieved. So this is the left hand side. You can see this is a normal how can actually the different water molecules or favor in the binding of the ligand in the active site. In the right hand side, there is a, depending upon the size of the active site and the size of the ligand, the binding interaction, strong or weak binding interaction involved. So here, in the first scenario, that you can see the ligand or receptor binding here. In the below B and C, you can see the active site size shrink. So that may be actually affect the proper formation of strong bond. In the other case, in C, the active site is fine, the size of the active site is fine, but your size of your ligand. So if your size of your ligand is higher than the active site or almost similar to the active site, that is also influence the proper binding at the active site. So these are the few parameters or few factors those involved, those are associated with the binding interaction. But beyond that, there is a lot of binding interaction and problem or binding interaction influencer or like that. So that is one of the parameter to select. So when we have to perform the docking, we have to check how much binding interactions are formed. Among these binding interactions, how much are strong, how much are weak, how much hydrogen bonding interaction, how much hydrophobic interactions or any other kind of weak interactions. And then second parameter is the scoring function. We can score the our different poses. So as I told you earlier, that is during the docking, search algorithm investigates the vast number of conformations for each molecule. So in active site after docking, a large number of conformation of the same molecule may happen. So among these conformation, we have to find out the best one or near to best one. So this scoring function helps to identify this. So scoring function may be used for the evaluation the quality of the docking poses. It also helps to find similar pose as the experiment orientation, that is how in the crystallization, how the ligand pose, which is the best pose in the experimental approach or the crystallization. So in docking, we try to find out the similar pose, and the scoring function helps for this. Also, we can identify the active or inactive conformation, or active or inactive compounds through the scoring function. It also helps to prediction of the absolute binding affinity, which pose more affinity to bind, which is less. That is also helped by the scoring functions. So there's a lot of scoring functions. So most widely used three scoring functions I'll explain here very briefly. The first one is the force field based scoring function. So here there is two parameters. One is called the bonded parameters, another is called non-bonded parameters. So bonded parameters like the bond length. So during the conformation analysis, how the bond shrink or expand, how the angle bend or expand, how the torsion angle change during the conformation analysis. So these three terms is called bonded parameters, and the another two parameters are non-bonded, like van der Waals and electrostatic interactions. So these bonded and non-bonded parameter of the each molecule play important role to achieve the perfect or the important orientation or conformation at the active site. So this is actually the force field based scoring function. So DOC or DOC-THROG, these are the examples. Another is the empirical scoring function. It is the sum of the various energy terms like van der Waals, electrostatic, hydrogen bond, dissolution, etc. And it is actually Ludy, Plants, or Glide score. These are the examples. And the knowledge-based scoring function, and this is basically the statistical analysis of interacting atom spheres between the protein and ligand. And it is, for example, drug score is one of the examples. So there is some more scoring function, but these are the widely used by the different docking engines. So if I sum up about this docking, that is, what are the steps? So the first, we have to define the structure of the target molecule. We have to identify the binding site. We have to identify the ligands or small molecule to bind. And then through the docking engine, we have to find out the binding mode. And then once we have the docking, then go for the docking analysis. So what are the applications in molecular docking in a different way? So we can go for the virtual screening. I'll explain shortly about how can we use the molecular docking in virtual screening. Target fishing, that is, you have ligand, you don't have the target, you don't know target, so that can also you can identify through the docking. Prediction of the adverse drug reaction, that is side effects. Also, you can use for polypharmacology or drug reproposition or ligand target binding rationalization. So these are the main applications of the molecular docking. So I'll go for the virtual screening today. Before that, another very much important application of the pharmacoinformatics and drug discovery is like design. So if you remember the slides, I explained when you don't have the known small molecules, but you have the three-dimensional structure of target. So that scenario, you can design your molecule based on the active site. So suppose this is your active site. So ligands unknown, target known. So this is active site where your ligand go bind. So based on this active site, you can use the different thousands of fragments to dock it. So suppose this is the one fragment, I docked it. You fit in the active site, and I find out which fragment or which position this fragment bind properly or fitted properly. So that position I select. Similarly, the other fragment I fit in this pocket similar way. So number of fragments, we one by one, we docked it, fit it, try to find out, sort out the different fragments. So suppose finally, among thousands of fragments, these 4 fragments we found in a different 4 places perfectly fit. So these are the individual fragments. So we have to make the complete molecule from these fragment. So we can join this. So joining is arbitrary. That is required the expertise. How can you hold this position? We need the single bond or multiple bond or any other rings to hold this position. So this is actually joining. After joining, we have the complete molecule. So this is one of the example for the hepatitis C virus helicase, and this is the first determination, the first find out the de novo design based molecule. Once you have this molecule, then you can go for optimization. So this molecule, this is structure first used by League Builder, one of the very crucial tool for the de novo design, and then optimize. So this is the second position, and finally you got this molecule. So this molecule developed from the de novo design. So this is another approach. So now I have explained the pharmacoinformatics. I have explained about the ligand-based approach like pharmacophore I have explained the receptor-based drug designing like docking, and another case the de novo design. Now I am going to explain the virtual screening. How can we use these pharmacoinformatics approaches or pharmacoinformatics models to search the search the small molecular databases. So first of all, I will explain how can we use the docking approach for virtual screening. So first of all, I need the structure of target, for example, protein. Then we need identify the small molecular databases, maybe PubChem, SNX, or Zinc, MQL, Mebris, etc. There are plenty of database. There is a large number of phytochemical database as well. So once we have this protein, once we have this small molecule, so we can go for molecular docking. Identify the target active site and go for the molecular docking. So each molecule will dock in this protein molecule and it will give the score. So once you have the score, based on the score, you sort out your molecule. Suppose I need the top 10,000 molecules. Among 1 million, I need the top 10,000 molecules. So we need the 100 molecules. So depending on your criteria, you can select some molecule. Sometimes we use the control molecule. Already known drug molecule is there or known standard molecule is there. That also will dock, and we compare the binding energy to sort out our molecules. So then you can use the number of criteria like drug likeness, binding pose, pharmacokinetics analysis, etc. And finally, you have a very less number of very less number of molecules, and then you go for the in vitro or in vivo experimental validation approaches. So here two very important things you have to keep in mind, that is selection of the ligand database, that is based on the criteria you are using in the docking or virtual screening, there is a chance you may slip your good molecules. So that is very much important. And molecular docking, even if your database is full of good compounds, if you are not able to correctly dock each one, you will have nothing at the end. So based on a selection of the docking engine, your docking parameters, etc., that is very much important, so that you can get the good molecules. And finally, you have to set up your criteria to rank the molecule. That's also important. If you are in error in the ranking, so you may lose good molecules. And this is very briefly the Pharmaco-4 based virtual screening. Once you have the Pharmaco-4 model, you can submit in the molecular databases, and then these model will go to the molecular database and fit each of the molecule, and it will retrieve some molecule based on this Pharmaco-4 model. This is called the initial hits, and then you can use a number of criteria. So these are the common, but you may have the other criteria depending upon your research project, and then you have to select the best molecule. So these type of finally the promising molecules you may have. So these are the application, and these are the overview, the different pharmacoinformatics approaches in drug discovery. So beyond this, there is a lot. Just these are the widely used and people are using. And now I'll go to explain the different application of the pharmacoinformatics approaches in the COVID-19. So the first example is the identification of active molecules against the NSP15 of the SARS-CoV-2. So what we did here, we consider the crystal structure of the NSP15 from the PDB, and then we consider the Acinix antiviral database. It contains about 9,000 molecules, the Acinix antiviral database. And then we went for the high throughput virtual screening molecular docking. So here actually there is a concept in the Schrödinger glide module, that is a virtual screening workflow. So this is 3 levels of docking: the HTVS, the high throughput virtual screening, then standard precision and extra precision. So we went for the 8,722 molecules for the HTVS molecular docking, and we consider we applied some percentage of the best molecules, and we considered about 2,500 molecules. And then we went for the standard precision docking, and we consider the best 10% of the input compound. So here we consider 261 molecules. And then finally we go for the extra precision, again with 10% of the best molecules considered, and consider 26. So just before going to the further, I would like to explain what are the difference between the HTVS, SP and XP. So all are 3 the docking approaches, but depending upon the conformation analysis. So in a HTVS, the conformation analysis are less. In the XP, but higher conformation analysis higher than the HTVS, but less than the XP, and the XP is a very exhaustive molecular docking approach and the high level of conformation analysis. So from after glide extra precision, we got 26 molecules from the beginning, and then we go for the binding free energy estimation using the MMGBS approach. And we consider 20 molecules, and then we go for the in silico pharmacokinetic analysis, and we found the 5 molecules actually satisfy the most pharmacokinetic analysis, and then we go for the molecular dynamics simulation, and finally the binding free energy analysis from the MD simulation trajectory. So these are the 5 molecules we found important for NSP15s of SARS-CoV. Then we check the binding interactions between this ligand and different amino acids of the NSP15. We found a very crucial few crucial interactions, and then also we try to find out how our ligand fitted at the active site. So we can see that perfectly all the molecule fitted. If you see in the last figure, the whole molecule together fitted perfectly at the active site. And then we go for the molecular dynamics simulation. So molecular dynamics simulation here, we generate the thousands of conformation of the protein ligand complex. That is, virtually we try to create the dynamic environment of the complex. because ultimately when it is formed as the medicine, it will go to real system. The real system always the protein and ligand are in dynamic states. So we checked it. Here we try to understand during the dynamic state how my protein and ligand are stable. So this is the RMST. RMST fluctuation indicates if it is less variation, so that it explains the stability. We found it. This is the RMSF, root mean square fluctuation. This is explain how the individual amino acids of your protein fluctuated in comparison to the native structure. So there is also no abstract fluctuation. This is the radius of gyration. This explains the rigidity of your system, how rigid during the dynamic state, and this is the binding energy of the each frame during the molecular dynamics. So you can see here the high negative binding free energy found from the molecular dynamics trajectory. And this paper just accepted last month, end of the last month, and online available. The second example for the TMPRSS2, and this is actually cell entry inhibitors, because TMPRSS2 is the host cell receptor, and this helps to cut the spike protein and ACE2 conjugation. So here we use the homology modeling because the crystal structure of this is not available. So we went for homology modeling to generate the 3D structure. We get the went for the active site identification, screening the molecules, and binding interaction analysis, and finally molecular dynamic simulation. So in homology modeling, we consider the sequence from the NCBI. We use the template from the 3D protein data bank. We went for the sequence alignment and modeling the 3D structure, and then after modeling we went for the loop optimization, sidechain modeling, and finally the model optimization, and then we went for the model validation. So we use the different tools like molecular dynamics, Q-mean, HotSIP, etc. for the model validation. So this is the final generated three-dimensional structure of the TMPRSS2 through homology modeling. And these are the different approaches we went for the validation of this model before using the molecular docking. And then we went for the two tools. One is the sitemap and another is the molecular operating environment tool to identify the active site of this protein. And we found these are the amino acids are present around active sites. So these active sites we consider for molecular docking. So in this study, we used for 5 standard drug molecule as a control. So we went for molecular docking of this standard molecule, and then we found these ambroxol and chemostat are the most important for this target and considered for the control in this study. So we use the SelectChem database. It consists about 2,000 molecules. We go for the homology modeling that explained. Then again we go for the virtual screening workflow. First high throughput virtual screening, then standard precision docking and XP docking. So after HTBS we consider 50% and SP 50%, and finally XP 25%. So these percentage of selection depends on you. How can you select actually? And then finally we got compare the binding score of the standard control molecules and these molecules, and finally 5 molecules. These 5 molecules found best molecule for this study, and then go for the dynamic simulation in silico pharmacokinetics. So these are the DOC score. I'm not going to explain details about this. This is the binding interaction analysis. The number of huge number of binding interactions we found, and here we see how the final molecule bind at the active site. So here we check the stability between the protein ligand through the molecular dynamics simulation, the RMSD, RMSF, and the RMSD of the ligand experience. So first the RMSD of the protein backbone, then RMSF and the RMSD of the ligand explain that the protein ligand complex were stable during this molecular dynamics simulation. These are the hydrogen bond analysis between the protein and ligand. And this is also published in last year. And the final example I would like to explain here, the screening of spike and NSP15 inhibitors together. So we consider some bioactive molecules. So here we parallelly consider the two receptors, one from the spike protein, another for the NSP15. And we use the same approach for both the targets for these molecules. I think about 100 or 145 molecules were there. And then we used some criteria like binding energy should be less than -7.4 depending on the control molecule. And then for the spike protein, we got 2 molecules. And in case of the NST15, we got the 5 molecules. And then we go for molecular dynamics simulation, RMS, RMS, etc. analysis. So these are the 5 best molecules we found for the SARS-CoV-2. So these are the binding index again, and how these are fitted in the active site. So here, I think this is for the spike protein. This is for NSP15, and then we go for the molecular dynamics simulations, and this is also published last year. Beyond that, our research group or collaborator also published other work in a different journal related to the COVID-19 recently. So I think I'll complete within, I have actually 2-3 slides are left. So now I'm going to conclude this study. So as I told you, that is the drug discovery process is a very complex process and time-consuming process. So how the pharmacoinformatics help to this solve the problem? And then different approaches are there. So what are the success story of pharmacoinformatics? If you look in the literature, these are the molecules. These are the drug molecules are available in the market. Those actually came in the market with the help of pharmacoinformatics or computer-aided drug designing. So list is, I actually this is the world data. So list is long, very long. And then finally, I like to conclude that the pharmacoinformatics actually widely used by the academicians and industry people. A lot of tools are available. So if you have fund, very good, then you can go for the commercial software. If you don't have, so no need to worry. There's plenty of freely available tools are there that you can use for your project. It is very, very easy to learn, very user friendly, and also very importantly, it is easy to roll back. Suppose in a certain time you go and found that is you are wrong, so you can easily roll back, but it is difficult for experimental approaches. It is reduce the cost, and it is very helpful to enhance the drug discovery. So finally, I would like to thank my supervisor, Richard Bryce at the University of Manchester. I would like to thank Dr. Sajal Kumar, Dean in the Adams University. Especially, I would like to thank Dr. Partha and Dr. Arindam Maiti for their help and invitation. And beyond all, I would like to thank all the faculty members, all the students of this Adams University to attending the conference and listen to me. So finally, I'd like to introduce my research group here. So this is our research group. This is my supervisor here. So if you go to this page, so you can see this current PhD or postdocs research group in the University of Manchester. And finally, I would like to thank everyone. Thank you very much.
Thank you, sir, for your wonderful and excellent lecture. It is, I am confident that everyone has gained knowledge and will apply in their research. And the students will also have developed some interest about the topic. Now, the question session is open to all. Uh, if anyone has any questions, please unmute and ask, sir.
Hello, sir.
Yeah, go ahead.
Sir, you are in which degree?
Oh, basically, I did my graduation in chemistry, then masters in bioinformatics, and then PhD in the drug designing. Now I am
Oh,
working on this
that
same
means
field.
you have PhD in drug designing.
Yes.
Oh, sir.
The degree is
like
not a matter if you are in the pharmacy or any field, any kind of science. Maybe nowadays I can say docking is very easy tool, and anyone can use, even the biologist or whatever. So degree, I don't think it is a matter.
So I just like to add with Atul sir that what sir said is a very important thing. That one thing you have to need, that basic infrastructure like the computer and the related software.
Yeah.
Although there are some license issue, but you can access some free software by which you can learn something related to these things if you are interested. That is the thing. But I think you need a very good workstation. So that is the most important part. Otherwise, you cannot do this type of work.
Yeah, that you are right. But if you don't have the, because for molecular docking, the normal computer is enough. But if you go for the dynamics and all these things, so the normal computer is not enough. So in that situation, there's a lot of servers are there. A lot of, you can hire those servers, and you can perform the molecular dynamics. And if you have budget, because computers nowadays very cheap. So if you have budget, so you can purchase your own system.
Right. So for the participant, if there is any question related to the presentation, so you can ask. So one question from my side, Atul sir.
Yes, sir.
Suppose we are doing some molecular, and after that we just find some specific target molecule.
Yeah.
Now this is available in a, means you can say available from market, like anyone synthesize it, or you can available from very known means company like Merck or any other company. Now, if after docking, if I go for then again some characteristics for that, if that drug is not available, and the synthesis is a little bit complex.
Yeah.
For that, if someone is not synthesizing this properly, because you know that chemistry is always always properly you can done in lab.
Yeah,
so
yeah.
For this, if there is any modification, we can done that to easy, that means that through the docking, that in related to the known activity. Known activity means near to that activity we can get by any easy molecule. So that type of procedure we have.
Yeah, definitely. That I told you. That is, after docking, you can sort out some molecules. Okay, so and then to improve the affinity or to improve the binding energy, you can modify the molecule. So as my experience, only the molecular docking is not sufficient to optimize the molecule computationally. For that, you have to go further. So you can go for the in silico pharmacokinetic analysis. That's one. You can go for molecular dynamics if you have infrastructure, or you can go for the quantum mechanical approaches to find out what are the functional group important for the reactivity and all these things. So based on this data, you can optimize. Trial and error method. So these tools will actually guide you what are the functional groups might be important, what are the not. So no software tool is available that is it will cook and give you to do for it. No, it will give some idea. Based on idea, you can optimize it, you modify it and recheck it. So that is the only procedure.
And
so
another thing,
yeah, go ahead.
Another thing, can we implement the artificial intelligence through the molecular dynamics or molecular docking?
Yes, yes. now, if I say that is artificial intelligence or machine learning approaches are widely used in the drug discovery. In case of the docking, in case of the virtual screening, in case of the dynamics or mechanics. So now, if you explore little bit, I think last 2-3 years, people are diverting in the molecular, the machine learning approaches and improve, because in early day, if you go back 5 or 6 years back, the people are trying to dock the small data set, 100 molecules, maximum 1000 molecules. Nowadays, people are using the millions of docking, and that is only possible due to the artificial intelligence or machine learning approaches. You have the machine learning approaches, you have the computational facility, GPU. So that if you have a GPU, that is very much easy to dock billions of compounds in a very less time. Because we use, I have, and I think last year, I docked about 0.4 billion compounds, and it took about 1 week. So if you have the infrastructure, so that is very artificial intelligence is widely used right now.
So that means it can be more flexible in the diversification of that research, and
yeah,
also
definitely,
and the less time consuming.
Your chemical space is large. Instead of a small chemical space, you are using the large chemical space. Your diversity of exploration of the molecules are huge. So definitely it there is a chance to get the good molecule.
Okay. So, another just to as it is very interesting for me.
Yeah.
So can you sir give me one highlight related to this, that how we can approach through the artificial intelligence process in our molecular docking or physical activity related thing? Just is a small example. I do not have any idea on that. So that's why
I think you asked the question very wrong person, but I'm trying to explain, because I'm also exploring this field right now. So actually,
is any coding or like thing?
Yeah, definitely. You should have the coding.
Then my question is wrong.
Yeah, Python is very powerful programming language for the machine learning. So if you have the Python concept, Python scripting expertise, so definitely you can use the different machine learning approach in your drug discovery.
Okay, so if anyone want to ask any question to Atul sir, please go ahead.
Hello, sir.
Yeah.
Samrat Singh here, sir.
Right.
I have a doubt in Pharmaco-4. Sir, actually, as we consider two main molecules, lead molecule and hit molecule, for clinical trial, for drug molecule discovery. Sir, actually, in lead molecule, as we consider molecule that have property similar to that of drugs. So, for in heat molecule, is it same processor that heat molecules properties, molecule of heat are similar like that of drug molecules?
No. Actually, here, if you are talking about my example, that I shown the example based on the known molecule, that is what are the functional group we are trying to find. But this is not the case when you are doing the pharmacophore model. So you have the diverse kind of molecules, and you know these molecules binds to that certain target. It is already known, experimentally proved. So you consider these known molecules and try to find out among these known molecules what are the functional group crucial for binding interactions and their optimal arrangement in the three-dimensional space. So we are not exploring the pharmacophore model based on the standard molecule or drug molecule. We are considering the raw molecules. Just we know these molecules bind to certain target, and then we consider those molecules to find out the best pharmacophore model. And once we have the pharmacophore model, that can be used for further screening or synthesis of the new molecule. So if you are talking about this, my example, this is example to just explain what is a pharmacophore. I think I hope the question is answered, Sabrat.
So, Dudu sir, please.
Any more questions from participants? Okay. Now I would like to invite Dr. Arindam Maiti, HOD of SOPT, for the vote of thanks. Over to you, sir.
Thank you, sir. So, first of all, I like to thank our honorable speaker, Atul sir, to take his time, busy schedule, take his time to make a presentation in this webinar. I like to thank first Dr. Parthora to arrange this nice webinar and contact Atul sir and discuss with him related to the time and all these things. I also like to thank Jadunandan Dey to arrange this seminar nicely with all the technical details. I also like to thank our IT man, that is Saptoshi Sannal, one of my faculty members, to arranging the whole thing, because the technicality, maybe I am not the person, but he and Jadunandan sir is actually arranged the whole thing. Last of all, I like to thank our honorable officiating dean sir, Dr. Sajal Kumar Jha, to give us the platform for this arrangement. I also like to thank our management, Chancellor sir and Vice Chancellor sir, to encourage us to conducting this type of webinar for both for participants and as well as for our students. And lastly, I like to thank all the participants for their gracious presence in this seminar or webinar, you can say, and their proper means interaction. So thank you all. Thank you, Atul sir, for your valuable time. I hope that maybe near future we can meet means physically in our international seminar or national or international seminar, as well as other presentation or other research related thing. I also like to encourage our faculty members, if they are interested to related to some research related molecular docking or dynamics or related things. So I hope Atul sir will collaborate with our faculty members in near future. And I actually wanted to or request you to make a combined presentation or means combination research work related to that. I hope it will be a very good for us for your valuable help. So thank you, sir. Thank you for your nice presentation. And I hope I end this webinar here, in this session here. Thank you all. Thank you.
Okay, thank you so much.
Please leave the meeting. Thank you.